At CM Bio, we've developed a range of single and multi-organ chip devices. And these are allowing for the first time the study of complex biological systems where previously such experiments would have only been possible with an animal. And these are cell culture devices that help us to predict the human response to drugs and other chemicals in the laboratory. Culturing primary human cells and stem cells in the laboratory is a very difficult and labour-intensive task. These cells are exquisitely sensitive to being outside of the human body. So a real advantage of organ-on-chip technologies is that they help us to recreate the dynamic conditions that cells experience in the human body. So for instance, changing hormone levels can be represented in cell cultures using organ-on-chip devices. In contrast with many microfluidic cell culture technologies, we've sought to develop a really streamlined product that gives the user an experience much closer to working with a standard microtiter plate.
With the different plate formats, I can set up experiments with different cell types or even with multiple cell types. And as an immunologist, I can use the microfluidic control to bring immune cells into contact with other tissue types. Different organ chip formats allows me to set up either single or multi-organ experiments with different cell types in different combinations. The organ-on-chip plates can be set up in any standard cell culture incubator and are linked to a single piece of hardware that controls the automation of the microfluidics. The OpenWell design of the device allows me to access the cells and the media throughout my experiment for the purposes of analysis. One of the great advantages of these organ-on-chip devices is that all the same techniques can be used with these devices as is used in standard 2D multi-well plates. This makes it really easy to train colleagues and introduce new users to the technology.
So in the last 5 years, CM Bio has worked with more than 25 pharmaceutical companies. And what we've done is we've concentrated everything that we've learned from these relationships with users of our technologies into our new products.